Welcome, Brett Johnson, OneMedRadio, New York City. Today I'm with Dr. Eugene Seymour, who's the Chief Executive of NanoViricides, symbol NNVC, traded on the bulletin board. The company's developed some innovative technologies for the treatments of viruses. Thanks for joining us today, Dr. Seymour.
Certainly, my pleasure.
So you had a big news this past week, the issuance of a patent that had been in application since 2006. Can you talk about the significance of that?
Certainly. I think this is really the biggest thing that's ever happened to us, because what it does is it really says that we now control the field of antivirals that are related to the use of polymers in the nanotechnology arena. And so there was no prior art. Dr. Dewan, the inventor of the technology, has been working on this for the last 20 years. Patent application was submitted some 6 years ago. And we submitted, I think, 31 claims, and we got 31 claims approved. The patent application is extraordinarily complex, but essentially what it says is you have this degree of protection, and this makes the company extremely valuable to a large pharmaceutical company because they really do like to have patent coverage.
So why don't you tell us in layman's terms, what does the technology do? How does it, in simple terms, how does it work?
Okay. In a sense, Brett, it's very elegant and easily described. What happens, of course, happens at a level that is, 20 thousandths of a meter in size, so that is quite complex, but I can explain it rather simply. Every virus needs a target cell because viruses are different than bacteria. Bacteria are self-contained. They are able to reproduce themselves. They have metabolism, and they don't need a cell. A virus is nothing really but containing of computer code, RNA or DNA. And what happens is the virus looks for the target cell, injects in the genetic material, and the genetic material hijacks the replication machinery of the cell to make copies of itself, 1 to 1 million copies. So there are some antiviral drugs. They try to prohibit the entry of this genetic material into a cell, and there are some that try to impede the exit of the newly formed virus created within that cell to the outside. We do things in a lot simpler way. What we have created is a nanoviricide. Nanoviricide is about 20, 30, 40 billionths of a meter in size, extraordinarily small, much, much smaller than a virus. Now, on the surface of the target cell is a protein, and that protein is the signaling protein. We call it the receptor protein. On the surface of the virus is the attachment protein, and when the two meet up with one another, they link. The virus is then pulled into the close approximation of its target cell, and it injects in the genetic material, which then takes over and replicates within the cell. What we do is we take that receptor molecule that's sitting on the surface of the target cell, and we make a very small copy of it. Just like to recognize you, Brett, I don't need your arms and your legs and all that other nonsense. I only need your eyes, your nose, your ears, your mouth, and then I can recognize you. It's the same thing with us. We make a very, very small mimic of what's expressed on the surface of the target cell of the virus, and we place that on the nanomicelle. Now we have the nanoviricide. And what happens is and we flood the body with trillions of these. And there are many more of our nanoviricides than there are of the target cells. And what happens is the virus attaches to our nanoviricide, which then opens up like a Venus flytrap and begins to encapsulate the virus, sort of sliming it, very much like Ghostbusters. And as that happens, it completely encapsulates the virus, and then through some complex physical-chemical interactions between our nanoviricide and the envelope, the covering of the virus, there's breaching of the wall of the virus, and outstreams the genetic material where it's picked off by the lowest level of immune system cells in the body. And then it's excreted, and that's the end of the game. And that's why we've had such incredible success, because we have this working, this system working, and working very, very, very efficiently. And, by the way, we can build this for any virus.
So what do you see as the principal applications of this technology?
Well, let's look at some of the more common viral diseases that have market shares in the billions. Influenza, and our nanoviricide for influenza will destroy any influenza A. And we've tested many, many different ones. We even tested H5N1, the bird flu virus. We tested that in Vietnam when they were having an outbreak some years ago. HIV, and we reported some very good results in animals with HIV. In fact, we reported a functional cure on 2 separate occasions, which means that you reduce the level of virus in the body to such a low level that you allow the antibody-producing cells to recover and control the remainder of the cells. And that way people can live a normal life, often, as we anticipate, with only 5 days of treatment and maybe an additional treatment every 3 months for the next year, and then after that, nothing. And they're not contagious because the amount of virus that they have in the body is really so dramatically reduced. And then there's herpes of the genitals, herpes of the eye.
And this technology is effective on all these viruses.
Yeah, all we do is change the receptor molecule. It's a plug-and-play. It's a Lego system. We just change the receptor molecule that's attached to our nanomicelle, and now we're set up for another virus.
So what's the status of the company? I mean, when will this be available?
Well, that you're going to have to ask the FDA. I suspect it's going to be available sooner in other countries than in the U.S., which is not an atypical situation. However, the clinical trials are cheap. They're quick, because influenza, for example, is a self-limited disease, and if it's over in 10 days. We're looking at very sick hospitalized patients, often those who are immune compromised, who have cancer, post-renal transplant, lung transplant, bone marrow transplant, or HIV. Mm-hmm. And these people have really nothing that can be done for them, and we feel that we can eradicate the influenza virus very quickly and effectuate a cure of influenza. Now, obviously, this has to be tested in humans, but from all the animal work, it certainly seems that way. And remember, the most important thing is, because you might say to me, well, wait a minute, you work in animals, big deal. It's the work in humans that counts. And in a way, that's true, but on the other hand, we're independent of the host. We don't care whether you're a man, a mouse, or a whale, or a dinosaur. We only care about the virus. And the virus is the virus. And it's the same in any of these animals. So we're independent of the host. And that's what makes this so amazing.
So it does sound truly impressive. So are you going to be in clinical trials now, or what's the...
We've had our first meeting with the FDA, and we're doing the things that they want us to do to prepare to file what's known as an IND, an investigational new drug application. And we're building a manufacturing facility to make this in volume for our clinical trials. So we're sort of gearing up, and I'm hoping that we can start work in another country much sooner, sometime next year, than we can in the United States, because if you don't need material produced in a special plant, that gives us like a 9-month earlier window to go and do the work in another country. And remember, for example, Australia has its flu season in our summer. So that gives us an opportunity to start looking at Australia as a potential clinical trial site. So we have the money in the bank to accomplish the task. We have the management, we have the drugs, and we have the consultants needed to help us get the manufacturing going. So we're in the best shape we've ever been.
Mm. Yeah, can you talk a little bit about some of the corporate side? You guys have been public now for a number of years. I know your stock has traded as high as $2.50 or even $3, and recently was down to $58, although it's come back quite nicely this last week as a result of the patent. Would you be raising more capital? What's the company's sort of capital situation and ability to get this product to the market?
Well, biotech companies always need capital. The difference between us and other biotech companies is that we're extraordinarily frugal. We have the drugs. We don't have to go through drug discovery. And all we have to do, really, is finish our toxicology work and then go into human trials. So will we raise some more money? I don't know at this juncture. I'm waiting to get some quotes. And as soon as I get some quotes for these trials, then I'll know how much they're going to cost. But the initial feeling is that we're really not going to need very much. If we do, it'll be a small amount. We've been public, as you say, 7 years. We had a high of $3.75, a low of about 10 cents. And now we're trading at 84 cents, giving us a value of, I believe, over $135 million as a market cap.
So why is now a good time to get into the stock?
Because we have this patent coverage, which is critical. We've had our first meeting with the FDA, and they've been very encouraging because there really is no good treatment for influenza. And, I'm a clinician, I'm a physician, and I've used the various drugs over the years, and unfortunately, none of them work very well, and they all have severe limitations. We're looking towards a cure for influenza with our Flucide, our first drug.
Interesting. Can you take us a second and tell us a little bit about your background and how you got involved in the company?
Yeah, sure. I'm a physician. I practiced medicine for many years, and this is my second public company. I was involved in the development of an AIDS test, a rapid AIDS test, used now in many countries around the world. And I met Dr. Dewan, the inventor, I think 9 years ago, and I thought, he's brilliant, but this is just crazy. And because no one was talking 9 years ago about nanotechnology. And over a 2-year period, it really, as I looked into it more and more, it really became clear that this was the future. This was penicillin for viruses. And I said to him, okay, let's form a company. We did 7 years ago. We backed into a shell. And like you mentioned, we've traded as high as $3.75 and also low, very low, right after we did that shell. But we're very actively traded. I think we've traded, in the last 3 days, 2.5 million shares. So there is a serious interest in the company, but they want to wait until they see human results, and then they'll start to talk to us seriously. By the way, we have, I think the last count is somewhere between 10 and 12,000 retail investors. We have no institutions involved, and we really don't do a lot of PR work. We go to conferences, and we're invited to conferences around the world, and we participate in many of them. But we've really been sort of flying under the radar. I think it's now time, because nanotechnology is enjoying a real boom, and people understand it. So when you say nanotechnology, they say, oh, yeah, I've heard of that. So I think the confluence of all of these events is going to continue to drive the value of the company. Plus, the work we're doing with one drug after another, and the idea is as soon as we go in to our IND with the first drug, the flu drug, we're going to have another drug every 6 months going into the FDA, because we got them lined up. I told you, herpes, adenovirus of the eye, dengue, HIV, and there's some others that don't have much commercial value, rabies, Ebola, things like that we've also done work on. So that's sort of the situation.
Well, congratulations. It's on the patent. It's truly an exciting story, and we'll be very keen to follow your progress. So thanks so much for joining us today.
My pleasure.
So that was Dr. Eugene Seymour, who is the Chief Executive of NanoViricides, traded NNVC. Brett Johnson, OneMedRadio in New York, signing off. Good day.